All right, how's everybody doing? We are working with metabolism today. So, what is metabolism? In terms of eukaryotic cells, I'm going to specifically relate this to animal cells. So, for us humans, metabolism is essentially just deriving energy from food. It's going to be a catabolic pathway, right? Catabolic meaning we're going from bigger polymers. We're going to use some enzymes, and we're going to break those into their smaller monomeric subunits. So for example, if we have a sugar here, we're going to break it down into pyruvate, and that is what we do with glycolysis. That's going to be the main focus of today. So glycolysis, like I said, is going from glucose to 2 molecules of pyruvate. Now you might be wondering, how are we doing this? It's going to be a 10-step reaction, basically. There's going to be 10 different reactions that go on. The whole process is 10 steps, and we're basically going from a 6-carbon glucose, so 6 carbon, and then we're breaking that down into 2 3-carbon molecules. So the main purpose of glycolysis is not to produce energy. That's a common misconception. The main purpose for glycolysis is to provide those pyruvate, which have high energy bonds within them, and also to regenerate NADH, which is essentially an electron carrier that we use in the electron transport chain later on to undergo oxidative phosphorylation and actually produce a great majority of our ATP that we actually use for the cells inside our body. So glycolysis itself, or catabolic processes themselves, are actually energetically favorable. Our body wants to go from bigger things to smaller things, which makes sense because this is the second law of thermodynamics, right? The second law says entropy is constantly increasing in the universe, right? So if you think about it, we're going to more disorderedness. We're increasing entropy by going from more structured to less structured, right? So if you think about it, if you're breaking down a protein, you're breaking the polypeptide chains, you're undergoing hydrolysis reactions, and you're getting, 2 dipeptides instead of four amino acids in a polypeptide. That is actually energetically favorable because you're going from one larger molecule to smaller molecules. Okay? So let's take a look at glycolysis itself. One interesting thing about glycolysis is that this has been preserved through all of evolution. Every single living organism undergoes glycolysis, whether you're bacteria or any other prokaryote, or if you're eukaryotic, if you're an animal cell or a plant cell, all cells that are living undergo glycolysis, which is pretty amazing. 3.5 billion years ago, bacteria were doing glycolysis, and this was when there was no oxygen present. So this is why glycolysis is an anaerobic process. It does not require oxygen to undergo this. But for animal cells, because we as humans, require so much ATP for daily activity, it's not possible to run solely on glycolysis or without oxygen at all, rather. We do have a mechanism for anaerobic respiration where we produce lactate, but still, it's not going to provide enough energy to actually, sustain a cell, at least for long term. So, these are the 10 reactions for glycolysis. We're going to go into these into great depth, but there's a couple things to make note of. One thing that's going to be critical is knowing your enzymes, or just classes of enzymes, rather. One thing to note that'll help all of this make more sense is the kinase enzyme basically adds a phosphate. So whenever you see a phosphate being added, you know it's going to be a kinase enzyme. If you could remember, which for me is the easiest thing, is to memorize the enzymes, and then we learn the actual intermediates later. Because that way you could kind of, if you can know the names of the enzymes, you know what's happening to each step. It helps make more sense of it. So kinases add a phosphate. We have isomerases or mutases. These basically change structure. So for these, we're not actually changing the molecular formula. All we're doing is changing basically the way that the elements are, basically the way that the compounds arrange. So whether we're changing a hydroxyl group's location from carbon 3 to carbon 2 or whatever, it doesn't really matter. So that's basically what an isomerase or a mutase does. And one other kind of trick that's really helped me out with this, as well as Krebs cycle especially, is the dehydrogenase enzymes usually, actually always, require an electron carrier. So for example, NAD plus is converted to NADH. So the NAD plus is going to get oxidized, and we're actually going to be reducing whatever is undergoing the dehydrogenase enzyme. So we'll see, for example, for us, that is going to be step 6 here. And another thing to note, so those are the main enzymes we want to know. Just memorize these, makes all of it way easier. Again, the dehydrogenase, all it's going to be doing as far as the structure is as far as the structure, we're just going to be removing one of the oxygens, and also dehydrogenating one of the bonds as well. But one other thing to notice is the net ATP. If you look here, we lose, so this is -1 ATP. This is -1 ATP. This is +2 ATP. And this is +2 ATP. So our net ATP gain is only going to be 2 ATP. Also, one of the most important things through glycolysis, as I mentioned earlier, is going to be the 2-NADH that we produce, because these are going to feed directly into the electron transport chain later on once we undergo oxidative phosphorylation. That's going to yield our 30 ATP, which is almost the entire cellular respiration ATP that's produced. So let's look at all of these in depth. I will explain to you guys, I'll let you in on a little acronym that I really enjoy that helps me kind of remember the enzymes. So for me, I remember this: happy, pirates, picked, all the greatest, picked, pumpkins ever picked. Now, how is this helpful at all, right? First off, it helps you remember, if you can memorize this exactly to a T, it helps you remember the number of enzymes. We know there's 10. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We see there's 10 there. Now, the secret is, if you look at the first letter, 10. So you look at the first letter of each of those, it tells you the first letter of each of the enzymes in each subsequent step. So the first one we saw here, So here, you see that we have a hexokinase. We have a phosphoglucose isomerase. We have PFK1 aldolase, triose-phosphate isomerase, G3P dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, pyruvate kinase, right? So if we look at our little acronym here, we can see we have hexokinase, we have phosphoglucose isomerase, we have phosphofructokinase 1, we have aldolase, we have triose phosphate isomerase, we have G3P dehydrogenase, we have phosphoglycerate kinase, we have phosphoglycerate mutase, we have enolase, and we have pyruvate kinase. Now another trick for this, once you memorize those, the key steps for this reaction regarding ATP production or consumption is something I like to remember as 1 plus 3 plus 7 plus 10, right? Ignore the pluses, that's just how I remember it. But the way I think of it is 3 plus 7 equals 10. We need step 1 and step 10, which is the first step and the last step. So step 1 and 3, so this step 1, and we have step 3. So we have phosphofructokinase 1 as enzyme 3, and hexokinase is enzyme 1. So that's 1 and 3. These both require 1 ATP. Now if we remember, there's 2 ATPs required for energy harvest. That means step 7 and step 10 are going to be our energy producing steps. So that is going to be right here. So step 7 and step 10. So that's going to be plus 2 and plus 2. Now why is it plus 1? Why is it minus 1 here and plus 2 here? Well, if you look back at glycolysis, you'll see that once we go here, so once we go from G3P, so glyceraldehyde 3-phosphate, we now have 2 molecules. So we went from the fructose 1,6-bisphosphate here, and through that aldolase enzyme, basically what that did is it broke up the 6-carbon molecule into 2 3-carbon molecules. That's why it's glyceraldehyde 3-phosphate. The 3 doesn't denote that there's 3 carbons. It's just the glyceraldehyde is the actual 3-carbon molecule. The 3 notates the position of the phosphate group, so it's going to be on carbon number 3. So that's where we get the ATP from, and that's where we get the 2 NADHs from as well, because there's going to be 2 molecules of each. So each one is going to be yielding an NADH, and each one is going to be yielding an ATP in step 7 and 10. But since there's 2 total molecules, there's going to be 2 total molecules of ATP produced and 2 molecules of NADH in those steps. So that's how I remember which steps produce ATP, basically. That's the easiest way for me to remember it. And then as far as the NADH, we know the NADH from what we went over earlier, what we know about enzymes, is the dehydrogenase enzyme is where the NAD plus becomes NADH. And we know, again, this happens 2 times. So if we go to our next slide, we'll go over the net yield. So net from glycolysis only, we have 2 ATP because we had minus 2 ATP plus 2 ATP plus 2 ATP equals 2 net ATP. Okay, so we have 2 ATP, we have 2 NADH, and we have 2 pyruvate. So, hardly any energy has really been produced yet. Like I said, most of the energy lies in this 2 pyruvate molecule, and also the 2 NADH, which are going to be basically, the energy is going to be extracted from those 2 electrons further once we get to the electron transport chain. Now the last thing I want to do is just to elaborate really quick on those steps in glycolysis, because it really makes sense if you actually look at this from a structural standpoint and from a molecular standpoint, what happens. So like I said, we're going from glucose. Okay, so if we look at these enzymes really quick, we go from glucose to glucose 6-phosphate. We need a hexokinase, because remember, the kinase basically adds a phosphate group, which is exactly what happened here. We still have our glucose. The only thing we did was we added to the 6th carbon a phosphate molecule. So where did that phosphate molecule come from? It comes from the ATP that was required to actually start this reaction. So ATP goes to ADP, so you have a net loss of ATP, but that phosphate gets added to the glucose 6-phosphate. So the next step we have is that G6P is going to go to fructose 6-phosphate. So what do we need for that? It's going to be the phosphoglucose, and then it's going to be isomerase. Because all we're doing is we're changing the structure. We're going from a glucose to a fructose. That's all we did there. So that's it for that step. Then the next thing we're going to do is we're going to do the PFK1, so phosphofructose kinase 1. And this is another step where we need ATP, because this is step 3, right? So what's happening when we lose that ATP? That phosphate ion that's being lost when we hydrolyze the ATP goes to the fructose 6-phosphate. So now we have fructose 1,6-bisphosphate. So the bisphosphate means bi, 2, and then the 1,6 notates where the phosphate groups are on the molecule. So we know that there's a phosphate on carbon 1 and on carbon 6. Easy enough? So then we're going to go from fructose 1,6-bisphosphate, and this is going to go one of 2 places. It's either going to go to the G3P, which is the glyceraldehyde 3-phosphate. This is the most desired product that there is. Or it can go to dihydroxyacetone phosphate, DHAP for short. Now there's an enzyme that catalyzes this reaction, and that's just going to be aldolase. So basically what the aldolase does is it's going to split that 6-carbon molecule into 2 3-carbon molecules. So this is why I said after this reaction there's going to be 2 G3Ps floating around. And to get from the DHAP to the G3P, it's going to be an enzyme called triose phosphate isomerase. Okay? So then, the next step, G3P, the next thing we're going to do, we need to get this guy over to 1,3-bisphosphoglycerate. So, what does it look like the enzyme here we're going to be using is G3P dehydrogenase. So what does the dehydrogenase mean? We're going from NAD plus to NADH. And this is going to happen 2 times. just for the sake of easiness, just remember every subsequent reaction following G3P creation or formation is going to be 2 times. So it's going to be 2 of the 1,3-BPG, there's going to be 2 of the 3-phosphoglycerate, and so forth. So this next reaction, we're going to go from 1,3-bisphosphoglycerate to 3-phosphoglycerate. Now, what happened? It appears as though we lost a phosphate, right? 1, 3, and now we just have 3, so we lost that phosphate group that's on carbon 1. So what do you think happens to it? You're right. ADP becomes ATP. So all we did was we attached the phosphate from the 1,3-bisphosphoglycerate to the ADP, and that gave us ATP. So now we have 2 ATPs produced because there's two of these reactions, and a 3-phosphoglycerate. That's going to be catalyzed by phosphoglycerate kinase. We are going to go to 2-phosphoglycerate. Phosphoglycerate kinase. Phosphoglycerate mutase, because a mutase just changes the structure. So we go from removing the phosphate group to here over to the 2-carbon. Then we're going to go from 2-phosphoglycerate, and then we're going to use enolase to go to phosphoenolpyruvate, and then the last step is to pyruvate. And the last thing we're going to do is use pyruvate kinase. And that's it. That's all we got to do. And again, this one is going to be ADP to ATP. So this is another ATP producing reaction. So in short, I mean, that's pretty much all you need to know. I mean, the key takeaway is that we have 2 NADH, we have 2 ATP, and 2 pyruvate that are produced. Now, with the pyruvate, we enter what's known as the citric acid cycle, the tricarboxylic acid cycle, Krebs cycle, whatever you want to call it. But basically, we're entering the mitochondria, because now the pyruvate's small enough to actually sneak into the mitochondrial membrane and actually be oxidized for energy. So that's basically, in a nutshell, glycolysis. Let me know if you have any feedback. Again, don't forget that acronym. Happy Pirates picked all the greatest pumpkins ever picked. Enzymes first, and then I would memorize where ATP is produced and learn intermediate names. And remember, this occurs in the cytosol and is anaerobic, meaning it does not require oxygen to occur. So thank you guys for watching. I hope you learned something. Let me know if there's any other topics that you wish for me to take a bat at. So don't forget to subscribe, comment, and like.